It's an honor to be addressing everybody at this international conference, and the first opportunity for me to share what our company is working on in this exciting field. So the title of the talk is 4D biomaterials, so what's new? Obviously, the beginning of the company name is 4D, and as everybody understands, that probably everybody understands, that refers to 3D printing, with which everybody's familiar, and the fourth dimension of time. And the way our materials behave over time is a key part of their interest. So what I'd like to do today is give you a preview of the talk. I'm going to start off by putting everything in context with a brief history of resorbable polymers, just to set the scene, and talk a little bit about how our founders started to work to develop a new range of degradable materials, degradable polymers that can be 3D printed. Then I'll go through the mechanical properties that we can achieve with these materials and their tunability, an interesting feature that they have, which is shape memory, and of course, say some important information about the biocompatibility data that we've established so far, and then talk about the tissue generation performance, which is a very important aspect of their properties, and then lastly, talk about the way that they degrade, and that's also another key feature of these materials. Lastly, I'll talk a little bit about potential uses, and then the position we're in the company, and the next steps we're hoping to take over the coming year. And that may, of course, involve some of you people, because we're very much keen to start working with collaborators to advance this technology. So a brief history of bioresorbable polymers. Having read up on this recently, when I started to work with the company, I was really surprised to hear that polylactic acid, one of the first bioresorbable polymers, was discovered in the 1920s by Carruthers working for DuPont. So these materials have a long history, and since the 70s, they were developed into commercial products, for example, resorbable sutures, and have built up a large market globally, which is in the region of in excess of $1 billion per annum, possibly up to about $1.5 billion per annum today. And the majority of that market is made up of materials like polylactide, polyglycolic acid, and polycaprolactone, and these materials. And they obviously have many strengths, which have made them very successful in these bioresorbable applications in the medical field. But they also have some limitations, which has inhibited the development of the use of these materials in certain applications in medicine. And one of the limitations is the ability to 3D print these materials in fine resolution into fine structures, and I'll speak about that in a little more detail. And the other limitation is the degradation behavior, which can cause issues in certain applications. So everybody can see that if they want to refer to it afterwards. It's obviously, we don't have time to go through it in detail today, but it's there for reference for people that need to see it. Again, this is a slide which my 3D printing advisor colleague developed for me, which you can refer to after the conference in detail if you wish. But this is a summary of the existing materials that are available today that can be 3D printed, but the limitations of the 3D printing processes are listed in that third column, that last column on the right. And I think it's all I want to do to summarize today is to say that whilst there are some 3D printing processes that can be used with some of these materials, they don't give you the resolution to produce finely detailed scaffolds with carefully constructed geometry. And that's one of the things that our founders were aiming to develop when they developed these materials that we're talking about today. So the unmet need, which our founders wanted to address, was that existing bioresorbable polymers cannot be 3D printed into flexible, porous scaffolds, and that their breakdown behavior can limit their range of use. In particular, well, I'll show you when we go through the detail, I'll talk about the breakdown behavior a little more depth then. So these materials that our founders developed, the founders, by the way, led by Professor Andrew Dove, who was one of the leading academics on the subject of degradable polymers, initially researching at the University of Warwick in England, and then at the University of Birmingham. So this work is the culmination of 12 years research. And the team developed chemistry around polycarbonate urethanes, and that's the sort of heading, if you like, of this family of materials. And they comprise, in the form of 3D printing resins, they comprise oligomers, cross-linking agents, photoinitiators, inhibitors, and all the sorts of things you put into a 3D printing resin blend to give it the shelf life you need and give it the 3D printing properties that you need. So this is an image of the actual resin that's been made in our laboratory in Nottingham, and an inset image of a 3D printed ellipsoidal scaffold model of a device that we're developing for an application that I'll talk about later. So first of all, these materials have a wide range of mechanical properties based on how we mix the blends together. We can make a range of materials that are from a very soft sort of modulus up to a relatively hard and rigid modulus. The materials we're showing you here today are the soft end of the spectrum. And you can see here two stress strain charts. On the left is compressive stress, on the right is tensile. So you can see that by these different lines are different mixtures of blends of our material. So you can see the one that's labeled poly-TM-PAC is the softest, and you can see very, very flexible materials. I'll show you an example here, which I can squash very easily with my fingers. This has very similar sort of modulus to adipose tissue. So we are developing that type of material for use in soft tissue applications. And again, in the model of an ellipsoidal scaffold, you can see it's very squashy. By mixing different blends together, we can tune those moduli to suit the exact indication that you're working on. And as I said, these are the softer variants, but we can also make a harder set, which takes the modulus up to in excess of 3 gigapascals, which is getting up to approaching the modulus of materials like PEEK, polyether ketone, which is used in quite a few medical implant applications. So this explains how we have quite a lot of control over the mechanical properties by changing the composition. The next feature that's very interesting for these materials and relates to the 4D time variant is shape memory. So these materials can be compressed at elevated temperature and then cooled and frozen into a compressed state. And then when inserted into the body, body temperature, they gradually recover a shape memory, but with very little expansive force. So if you look at the bottom set of photographs here, this is an alginate model, which is a commonly used material to model soft tissue for ballistic testing and so on. And a void has been created here to simulate a void left in a soft tissue after a medical procedure. And here is an example of our 3D printed material cube. which can be compressed, as shown in the example held by the tweezers, inserted into the void, and then at elevated temperature, i.e. above body temperature, seem to expand to fill the void. And on the far right, you can see after removal of that expanded piece of material, the void shape is virtually unchanged. So there's very little expansive force on the shape of the void during this behavior. And we believe that that will make the materials a very comfortable material to have implanted into the patient, because they will be far less palpable than more rigid implants that some patients have been experiencing in the recent decades. In the top line, you can see a rigid material with a hexagonal hole in it to simulate the behavior in a harder material like bone, and a version of our material which is again has been cut to fit into the and squashed down and frozen, and then allowed to fill back the hexagonal shape, and then to the right of that, irregular shapes. So this shape memory property we think can be very interesting and useful in a number of applications, and that's part of why we're called 4D materials, because the shape memory is part of the 4D functionality. The next very important consideration, of course, is biocompatibility of these materials. And initially, the materials have been tested in vitro in a wide variety of cell lines, both animal and human cell lines, and had very encouraging cytotoxicity data. This information I'm showing you here is based on some in vivo analysis that was done using 4 samples of material. The TMPAC and the NTC are 2 grades of our material that are in a 3D printed porous scaffold. And then as a control, we've got the TMPAC, our material, in a solid format, and the polylactide material, again, in a solid format. And this chart shows you here the collagen thickness of an encapsulation around these implants. So this is an indication of the foreign body response of the body to these implanted materials. And you can see here that after 1, 2, and 4 months, the polylactide control had a much greater collagen capsule thickness than both the solid material that we made, and more importantly, the porous materials, 3D printed porous materials, which show a much lower foreign body response. So again, we think this is very encouraging data for these materials. We acknowledge, of course, that biocompatibility is indication specific and even patient specific. So this will ultimately have to be tested in clinical trials, which we are planning to embark on in the coming years. So this is an interesting slide as well on the looking into the tissue response, again in murine in vivo studies. And the diagram on the right, you can see, if you see the honeycomb-like center, that's indicating the implant scaffold, and the 1, 2, and 3 are the places relative to the implant and the surrounding tissue where these sections were taken, and where the tissue infiltration and scores were marked. So row 1, which is the outside of the material, row 2 at the interface between the flesh and the implant, and row 3 in the middle of the implant. So you can see on the left-hand column, which is the adipocyte infiltration score, at all 3 points of the work, our materials, both the porous black and red and the solid blue, showed very good infiltration across all of the scaffold and the whole of the structure. Again, on the right, the fibroblast scores again, across all three situations, were very encouraging with our materials. And relatively, the controls for polylactide and to a lesser extent, our solid materials show that these 3D printed porous polycarbonate urethane materials have very interesting tissue response and the prospect of helping to accelerate healing in implantation applications. So moving on to the degradation behavior, we can tune the degradation as seen on the left-hand column here. This is accelerated data, so the time scale is not real time. This is acceleration, but the point that's important is that the gradient of those degradation curves, it shows how we can tune the rate of degradation by blending the materials differently. So we have this combination of blending the materials to tune mechanical properties and degradation properties. On the right-hand side of the diagram here, you see some in vivo data, which is obviously real time, again, murine studies, which show a polylactide control, which is the black line at the top, which shows that even after 4 months, measured by gel fraction, there is a very little degradation occurring. And we know from the literature and from the data from medical applications that we've looked at and feedback from clinician interviews that sometimes polylactic acid and polylactide and PGA materials like that can have a foreign body response which causes them to be encapsulated, and then the degradation is sort of held up whilst the bulk of the material degrades and eventually releases the breakdown products of lactic acid and glycolic acid into the surrounding tissue. So you sometimes see in the literature and in the history of these devices, especially when used in larger quantities, you can have, after some years, an inflammatory response and pain response, which has limited the size of these implants that you can make from these materials. In contrast, our materials are starting to degrade much more quickly, and they degrade by surface erosion gradually from the outside of the 3D printed structure in. So they're gradually being degraded and resorbed into the surrounding tissue at a much more sympathetic rate to the rate in which you want the tissue infiltration to happen. So what we're aiming to do here is to create tissue infiltration followed by degradation to provide the optimum healing response. So the potential uses for these materials are very many. Every time I talk to another collaborator, they seem to come up with another potential use. So for the broadly two main areas, soft tissue and harder tissue. So we are working initially on soft tissue applications, but we're also starting to develop our materials for the hard tissue applications. So bone related orthopedic type applications. And the first soft tissue application that we've really looked into is breast conserving lumpectomy devices. So this is where a woman that is undergoing treatment for breast cancer may have a tumor removed, leaving a tumor void space, and we will make a scaffold device such as the one I'm showing you on the screen here, with this sort of very soft compressible structure, and that would be during the time that the tumor is being removed, the surgeon would compress this, or his assistant would compress this, and to the required shape, freeze it at that size, insert it by minimally invasive techniques, and then that would gradually recover its shape in the way I described earlier to fill the void and to enable the tissue around that void to infiltrate and recover and heal as the implant degrades. And the work that we've done with key opinion leaders in working in this field suggests that this has potentially big benefits in helping to accelerate healing and good cosmetic outcomes for these types of procedures. So this is an area that we're doing a lot of work on, and we hope to, in a number of years' time, ultimately achieve a regulatory approval for such a device. And then in orthopedics, we've developed a concept for an implantable bone scaffold device that would be used in long bone fractures, such as this one here shown to the tibia, where a comminuted fracture, which is typically managed with large amounts of autograft material taken from the iliac crest or somewhere. That procedure in itself can be very painful and obviously add risk and cost. And we think that by if these materials work as we hope they will and believe they will in this type of orthopedic setting, they could be used to reduce or eliminate the need for autografting of bone material from other parts of the body. And that could be great news for both the patient and for the health provider. There's also the possibility that they could be used in combination with bone growth factor materials to make them more efficient and in terms of their quantity required and the distribution of the bone growth factor in the fracture location. So that's a couple of indications that we're working on, but collaborators are also working on, are interested in working in areas related to dental implants, micro devices for things like sutures, suture anchors. So there's a wide range of possibilities from these materials. Our current position is that we span out from, the company was span out from the universities of Warwick and Birmingham in May of last year, 2020. And I joined the company in April, just before that, to help make that happen. And we have now established an independent laboratory in Nottingham in the UK, which has enabled us to work through COVID because it's independent and now people can go in there with plenty of safe space to move around. There's enough space for about 8 people in there and we've got 3. So it's a great facility. And we have a resin synthesis and blending capacity there of with 10-litre reactors, which can give us about 500 to 1,000 litres a year, which for R&D purposes is a great step on the journey to scaling up. So we can make 10 litres a week or so. We can use with collaborators to develop other device concepts. So one of the things from today is we're very hopeful that we will meet some potential collaborators that we can work with to help fulfill the potential of this technology. We are currently raising a seed investment round, which will help us to complete the preclinical development of the technology. So again, if there are any investors in the audience, we're delighted to speak to you. And lastly, I'd like to thank you for your attention. And here is my email address and our website. If anyone would like to find out more, you're very welcome to get in touch or visit our website, and we'd be delighted to speak to you. Thank you very much for your attention.